Hello everyone, and welcome to this quick walkthrough of the paper GenePT, a simple but effective foundation model for genes and cells built from ChatGPT by Yichun Chen and James Doe. Foundation models are nowadays designed to learn embeddings of genes and cells, which are essentially numerical representations that capture their biological properties. These embeddings can then be used for various downstream analysis, such as classifying cell types or predicting gene functions. Existing methods for creating foundation models such like Pharma and GenePT require extensive data curation and resource-intensive training. This paper proposed a simpler alternative called GenePT, which leverages ChatGPT embeddings of genes based on literature. GenePT used text description of genes from NCBI database and the powerful language model GPT to generate gene embeddings. These embeddings capture the meaning and the context of gene based on the information in the literature. GenePT then generates single-cell embeddings in two ways. So today I will walk you through this main procedure of GenePT with more details as what I also did for my own learning purpose. So the resources, the starting point of the GPT is text format of like gene description. This is usually from customer-defined reliable description of genes. So here the authors used the National Center of Biotechnology Information (NCBI) that centralize gene-related information into individual records. So here example of a gene named CD24. The next step is using the text description of the genes as input for GPT and generate high-dimensional quantitative embedding values of that corresponding gene. As example here, showing the partial of the embeddings of gene, because the embedding dimension of the gene is all like about 1300 dimensions. If you're interested, I will encourage you to watch the following up videos of step-by-step implementing this GenePT, as well as showing you with more details of this usage of the method. So with this quantitative embeddings of the genes, we can then conduct multiple downstream analysis, including gene clustering, gene-gene interaction, protein network, and functional prediction. So the way the authors built from the gene embedding to cell embedding is two ways. So the figure B actually describe one as average embedding of all the genes like expressed in the cell, and also another way is described in the figure C. So the authors can rank the genes is expressed in individual cells and then input the gene orders as a sequencing of sentence, which can then be treated as another test input for the GPT embedding model, which can generate a new embedding for each individual cell. So based on these two formats of cell embeddings, we could conduct a further like a cellular level analysis, including cell clustering, cell type annotation, etc. So the author shows using the gene embedding generated from the NCBI text and the ChatGPT, actually they could observe very well separated gene functions as shown on this UMAP on the left side, as well as authors did prediction tasks for the gene functions using the gene embeddings, and you can see in the confusion matrix shown on the right side, which showing very high accuracy of the prediction. And the other performance shown here is a protein-protein interaction prediction, which really showing that GPT is outperforming the other current existing foundation models, including SCGPT and the GeneFarmer, with different three different data sets, which covers protein-protein interactions. So overall, this paper showing using a simple but very effective way, you could actually use existing embeddings from the current large language model, not only ChatGPT, but you can also explore other like Gemini and other like current very effective language model embeddings to study the single cells as well as uh, proteins level understanding of either cells or the even like the patient samples. So I think this method is really inspiring, and I will encourage you to watch the following up implementation and exploration of the um, GenePT application in my following up videos. Thank you.